OsteoBites is a weekly osteosarcoma webinar and podcast presented by MIB Agents. Today we are talking with Dr. Damon Reed about Moffitt's osteosarcoma research approach. Our OsteoWarrior panelists are Ryan Kennington, MIB Agents Junior Board Member, and Amanda Levine, Patient Advocate. I'm your host, Ann Graham, MIB Agents President. Welcome to OsteoBites. The bite today at MIB Agents HQ is something I'm rethinking. It's baked garbanzo beans, and there's no chocolate. I'm not sure I'm going to eat them. Anyway, hope you have your snack ready because we have a very cool OsteoBites today with Dr. Damon Reed of Moffitt Cancer Center. Dr. Reed is going to be talking to us about their center's approach to osteosarcoma research. Dr. Reed is the director of the Adolescent and Young Adult Cancer Center's program at Moffitt Cancer Center, and he's the interim chair of the Department of Individualized Cancer Management. He's also the associate professor of pediatrics at the University of South Florida. There's so much more to tell you about Dr. Reed, but I'll just go to the best part. He is a member of the MIB Agent Scientific Advisory Team, which we are very grateful for and feel really lucky about. Our panelists today are OsteoWarrior and Patient Advocate Amanda Levine, and OsteoWarrior and MIB Agents Junior Board Member Ryan Kennington. I'm your host, Ann Graham, also an OsteoWarrior and President of MIB Agents. MIB Agents makes it better, MIB, for kids with osteosarcoma. We help kids facing this aggressive cancer by providing direct patient and family support and through educational programs. If you are on this webinar, you know that osteosarcoma is the oldest known cancer with some of the oldest known treatments. So we also support the researcher and physician community with an annual conference, through OsteoBites, and by funding meaningful osteosarcoma specific research. If we can be of help to you, please just say the word. Dr. Reed, would you get us started by introducing yourself, please?
Well, thank you for that lovely introduction. I would just say I'm an aspiring, hopefully I'm getting there, an eco-evolutionary oncologist, and that's what I'm going to be talking about. I take care of patients between the age of 18 and 40 with all sarcomas, especially osteosarcoma as well. My OsteoBite snack is what's sitting on my desk, some soft granola, blueberry cobbler. It's delicious.
Well done.
Hi, I'm Amanda. I was diagnosed with OS in 1987. Since then, I graduated college, I got married, I have 2 kids, and I became active in 2010 with the OS community when I was diagnosed with breast cancer. And I survived that as well. And 2 years ago, in 2018, I was diagnosed with ALS, which is why I talk funny. And it's so good to be here.
Hello, my name is Ryan. I was diagnosed with osteosarcoma in 2014. And I had a limb salvaging surgery. 1 year after I finished treatment, I had a relapse in my lungs, and I had a bat surgery to get that removed. And ever since I've been cancer-free. And recently I just graduated from college.
Fantastic, what a panel. This is very exciting. So at Moffitt, one of the exciting and unique skill sets that one of our departments has is we have an integrated mathematical oncology department and a cancer biology and evolution program. And these are led by people like Elsa Flores and Bob Gatenby, who is a radiologist down here on the right. You'll notice a theme of chalk and chalkboards in a lot of these pictures, and a heavy rooting in mathematics that drives a lot of these. I will not pretend to understand the mathematics, though I'm trying to learn some of it. Above that, you'll see a phylogeny and an I think, a famous kind of sketch by Charles Darwin, which also guides the principles of these things, of the strategies. And you'll see Joel Brown, an evolutionary biologist, who also is a machine of the ideas behind some of what I'll present today. we all have conflicts and we all work with companies and other things. I wear a lot of different hats. Today I'll be talking mainly with my Moffitt and my National Pediatric Cancer Foundation hat, but I'm very lucky to work with Factor MIB Agents. I know this is a little bit of an older icon, and many other places that support the work that we do here and myself, and I appreciate it. So first, as an eco-evolutionary oncologist, we always talk about environment and habitats, and so I work at Moffitt Cancer Center, an adult cancer center that takes care of patients 18 and older, and that's where I see my new patients, 18 to 40. And also have a role at Johns Hopkins All Children's Hospital, the Pediatric hospital, which is across the bay. This is the Tampa Bay area on the west side of Florida. And in between there is the headquarters of the National Pediatric Cancer Foundation, which is funded most of my protected time for the work that we do. In the AYA field, there was at least about 20 years ago this battle between two regimens. One was called Hypersea Bed, which is similar chemo over and over again for acute lymphoblastic leukemia. And there's a pediatric way of treating acute lymphoblastic leukemia that involves many agents over 3 years' time. It's much more complex, much more non-intuitive, and much more flexible. If you try to draw this out, the AYA field, there was at least about 20 years ago this battle between two regimens. One was called Hypersea Bed, which is similar chemo over and over again for acute lymphoblastic leukemia. And there's a pediatric way of treating acute lymphoblastic leukemia that involves many agents over 3 years' time. It's much more complex, much more non-intuitive, and much more flexible. If you try to draw this out, this bottom side is what ALL therapy looks like for a standard risk patient. It's a bunch of different agents given seemingly random. Some of them given over a long period of time, some of them given acutely over a couple days and then never again. And because of trials, because of the accessibility of leukemia, this 3-year standard of a lot of different schedules of agents has improved the cure rate dramatically. I think one of our biggest problems in sarcoma is that we have a Hypersea Bed-like treatment protocol. We start with some chemotherapy on day 1, and we keep doing it over and over again for 7 months. And I think that if I had to choose one thing that I would like to work on, I would like to figure out a path forward where our trials and the way we approach diseases like osteosarcoma are far more flexible, far more dynamic than what we do now. And so I think that because of the rarity of osteosarcoma, because it's harder to access, I do think smaller trials bringing in the opportunity for more ideas is important. So that's a general thought of how a big picture approach to osteosarcoma. How we practically do it at Moffitt is Moffitt has a really good clinical research infrastructure. You guys saw the MD Anderson talk on a previous OsteoBites, also talking about trials. And we work with this National Pediatric Cancer Foundation and work with hospitals across the country to collaborate, to be nimble, to bring new ideas, to continue to add sites, and to try to fund research that's going to lead to the next exciting trial, and then to actually conduct that trial. And there are institutions all over the country, and I just show this to show their logos and just demonstrate that it is a national consortium. And one of the trials that we run that this group would be interested in is a combination of Azacetine and nivolumab. It's a phase 2 trial trying to see if these agents work for patients that have relapsed with osteosarcoma and have the ability to have their disease resected. Patrick Thompson at the University of North Carolina and Mihaela Druda here at Moffitt are the principal investigators of this study. And what happens is patients receive 1 cycle of an immunomodulatory agent. They have their surgery, and then they go on to receive the same thing afterwards. And we're going up on dose levels. So this first dose level has been filled. We've shown it's tolerable, and we have patients that are continuing on treatment. That E is a typo that I now see very blatantly. And then we're going up to dose level 2 and 3, adding an azacitidine agent to try to see if we can stimulate the immune system a little bit more against these osteosarcomas. Again, since patients get resected to no disease, we're trying to really test if this particular immunotherapy in combination can eradicate small volumes of disease. There's already a bit of a signal that large volumes of osteosarcoma are not really affected by an agent like nivolumab by itself. again, trying to be team scientists and to share the wealth and to bring experts from all over the country. There is some biology attached to this study, just like all the studies that we conduct. And some samples of blood along the way go to Dr. Masanori Hayashi in Colorado. Other samples of blood go to Dr. David Loeb at Montefiore. I think these are names that you guys are familiar with. Ted Leitch and now Pat Levy are instrumental and in working with David Loeb to test the stool microbiome, because we all have bacteria living in our guts that affect the way we respond to immunotherapy. And so stool gets sent to Texas for patients. And the frozen samples of the resected tumors are going to Brian Lato at Johns Hopkins, and he's studying the immune infiltrate. And so I'm really proud of the fact that here at Moffitt, we're able to look beyond our own walls for osteosarcoma patients and bring in the best talents around the country to not only to give opportunities for patients to go on trials, but then also to make sure every precious sample is being taken care of by scientists that I totally endorse and believe in. And it's always wonderful to be able to work in this kind of team. I'd open the floor to any questions from the panelists of just this first kind of foray into what we do at Moffitt for osteosarcoma.
Thank you. And so exciting to know how many different cancer centers and oncologists are working together. And I think there's, I'm going to call it a myth out there that I heard many times when I was in treatment, that everybody's working in silos and nobody's sharing. And in the last 5 years with MIB, I have found that to be completely false, that the institutions like working with each other and they like to have ideas to bounce off of, and they really recognize areas of expertise that are all across the country. And it's like I think it's like anything. It's like with MIB, like we can't do everything under one roof. So we like to work with other nonprofit organizations and institutions, and it's the only way forward is together. So well done on that. Really excited about that. The other thing I had to say was a question, and that was, can you tell us more about the phase 2 trial for osteosarcoma? Amanda and I specifically will, if somebody asks for what's happening in trials, of course we send them to clinicaltrials.gov, but of course that can be complicated. So if there's something that we can point out, or we can have a place to send them to call, that's really helpful. So can you tell us more about that and where the trials are taking place?
Good. First, I'd love to take credit for the idea of working across the silos, but just like you, as advocates have led the way here. And so Melissa Helms was the founder of the National Pediatric Cancer Foundation, and she drilled it into my head in 2009 when we started. You must be egoless, you must be cooperative. We have pictures of us holding sledgehammers. You have to think beyond your own walls, and don't let that stuff get in the way. So while I get to represent this consortium, the ideas came from advocates, and so thank you for making that loud and clear that we need to work together. It's easy once you make that decision, then it's easy to be a meritocracy and say who's the best at doing this particular part, and then giving that person that part. So that's been all very easy. Now the trial, it is available. It is findable on clinicaltrials.gov, but I agree that's a tough system sometimes to navigate. Nationalpcf.org would have information. You could email me, damon.reed@moffitt.org for information. There are other contacts, but you can contact the National Pediatric Cancer Foundation. You could contact Moffitt to find this. The NCT number up here is how you look it up on clinicaltrials.gov, and that's a super helpful thing. So if you go into a Google browser and you just type in this, you can't memorize it, but NCT 036-28209, the study just pops right up. So the elevator talk about this is, if you're an osteosarcoma patient, you relapse to your lung only, doesn't matter if it's the first relapse or any, and the surgeon says they can get it all, you're eligible for this trial. So those are the patients. If the surgeon can't get it all, if the relapse is outside the lungs, not eligible for this study. It's a phase 2, which means what we're trying to determine is whether or not this works, whether or not this prevents this disease from coming back and hits a signal. It's the pre-specified bar trying to prevent relapses from coming back. That was so well laid out by Katie Janeway and Joanne Lagme in that paper in Journal of Clinical Oncology in 2016. And it's the same bar that is used in some children's oncology group studies. So 39 patients or so will go on this study, depending on how it's gone with the patients so far.
Okay. How long does a phase 2 last?
Yeah, it will go, this will be open for another 2 years, I would say.
Okay.
Yeah, I think there's enough of a need for this trial that it could, potentially fill within the next 12 months, but our sites aren't everywhere. And not every patient signs up for a trial when they're eligible. Some people use chemotherapy in these contexts as well. So those are the competing things.
Is there a limit to the number of participants, or?
Yeah, eventually we get up to a limit, but we're nowhere near that right now. So for the time being, we're ready to go. So the next, there will be 5 more people put on dose level 2, and then there'll be a pause to make sure that was tolerated for a month or 2. Then we'll just go from dose level 3 on.
What locations are the trials taking place, and are the meds oral or infused, and what are the side effects? Good.
It's IV. All these medicines are IV. Azacetine is given IV, sub-Q or IV over 5 days. So it'd be like a Monday through Friday, and then the following Monday and Tuesday. It has side effects of like some nausea and some low blood counts. Not like MAP chemotherapy though, but it can affect bone marrow and make it low in infections. And then nivolumab is one of these checkpoint inhibitors. So its side effects are generally immune-mediated, a hyper-vigilant immune system. So it could be colitis, some attacking of the colon, could affect some hormones, but generally these are rather well tolerated. But over a consent talk, we would go through the details a bit more.
Does CyberKnife radiation count as resection?
In this case, if a patient was saying, I have disease, it's in my lung, I don't want surgery, I want to do CyberKnife, they would not be eligible for this study. That's a good question. We're going to get a little bit more philosophical with the second part, and we're going to say, what is cancer? And so I'll open it up to the panelists. What is cancer? If you want to throw out some thoughts.
Cells going rogue in one specific area, in our case, bones.
Awesome. Okay, Scott, Ryan?
I'd say a wound that never heals.
Okay, good. So I was taught it was fast-growing cells growing out of control, cells that don't die. Both of you gave more nuanced, and I like it, kind of an idea of what cancer is. It's a wound, so you brought in some ideas of the environment, not supposed to be there, collection, those sorts of things. So that's great, and we're going to get to it. I generally clinically approach cancer with the idea of act like you've been there before, because I try to anticipate all the various things that cancer can do, and have kind of a pre-specified, at least starting point of what I would do. So if I had a patient, if the emails that we get, and if you get an email that says their patient had this big a tumor, it's located here, has this many metastases, or it came back this many months, I've written a paper with some other people I trust a bunch, and I generally follow that paper. So if people want to know what out-of-the-box things is Dr. Reed thinking about, I would say it's probably going to be my paper, is going to be where I would start. And then we kind of discuss from there. And again, it's like a group's paper. But, I try to kind of have an approach set ahead of time. Increasingly, though, once you get off that pathway, once things haven't gone the way that they should, I think of it this way. So this is the first bizarre slide that you wouldn't have expected to encounter. There's a dog and a wolf on top, and there's a squirrel and a tree on the bottom. Once a squirrel is alerted to a dog or a wolf, it'll run to the closest tree. A dog knows that. The dog does not know that, but a wolf does. I think of my job as taking care of the patient, number one, for sure. But there is a cancer that is out of control, a wound that won't heal, something that's grown, left the rails, something that, as Anne said, it just absolutely stinks and we hate. But it's doing something, and I'm doing something, and the patient's doing something. And one way of doing things is to react, and that's what a dog does. Goes after the squirrel, The wolf doesn't. The wolf says, I know what that squirrel is going to do, and it runs towards the tree. If you think about what the cancer is going to do, if you're thinking one step ahead, you have a better chance. I don't know that we all do that perfectly well in practice, and I'm still trying to live up to this. But the squirrel runs to the tree, dog ends up short, and the wolf gets the squirrel. And this is a paper that the evolutionary oncologist Joel Brown and Bob Gatenby wrote with our former CEO Bill Dalton and some others. And it's game theory. It's anticipating cancer. Wolf gets squirrel, dog ends up constantly chasing squirrel. Cancer reacts to what we do. And as soon as we give that first dose of doxorubicin and cisplatin, our first dose of AP, that cancer is different than it was before. I don't think of cancer as static. I don't think of cancer as a mutation. Cancer is a living population of cells competing with themselves, and it's adapting constantly. And as an evolutionary oncologist, cancer is a speciation event. It's when one of our cells decides decides is the wrong word, but when one of our cells is the unit of natural selection, it is the thing being selected for instead of our entire bodies, that's cancer. At the first time a cancer cell divides, I think of those two cells as being different. I do not think of every cancer cell being the same. I think of them all being different, but we have to group them in some level. We can't have 7 billion different cancer cells in someone's body and have a rational approach to that. In this case, this first cell divides and one is right next to the blood vessel and one is a little bit further from the blood vessel. They're genetically identical. Every single letter of their DNA is the same. But because this one is right next to the blood vessel, it's going to see more doxorubicin when the doctor gives it. And this one might not see as much, and it might live. And so not only the cancer cell, but where it is in the body probably matters a ton in terms of how it behaves. I am not a genetic person. I am not if we can just sequence every tumor, we'll figure it out. I'm a phenotypic person. If the position of the cell matters, how it would react to a stress matters, how it behaves matters more than its genetic code. So phenotype, where the cell is, how it behaves, how it can react to things drives the selection and whether or not that cell lives through chemotherapy. And after every single cycle of chemotherapy, the resulting mix of populations are better adapted to future strikes. And in this case, I just discussed that these cells, cancer cells, are better adapted to the human body than our normal cells are. That's why they steal things. That's why they grow. So terms. If I'm over in the IMO department and we're talking about cure, we instead talk about extinction, trying to make those populations become extinct. Instead of talking about genes like RAS or PI3 kinase or mTOR, which are genes you might have heard about, we talk about optimal foraging. Because all of those genes, all they're doing is trying to help the cancer cell get more sugar and more protein. And that's what animals have to do. They have to eat sugar and protein. When we talk about the Warburg effect, or how sugar is used by cancers, the principle for PET scans, that's speed of adaptation. When we talk about immunotherapy, we talk about predator-prey relationships, T cells trying to kill cancer cells, wolves trying to get squirrels. When we talk about how fast it can adapt over generations, it's things like loss of p53, another gene you may have heard about, and we call that evolutionary speed. But there are other cool things about ecoevolutionary and phenotypic approaches to cancer, things of limits of mutational tolerance. We all hear about how crazy osteosarcoma's genome is, but there are some genes that it can never mutate. And those are also vulnerabilities, but not too many people think about those. Importantly, we think of cancer therapy as natural selection. It's a way of selecting for cells, and that's probably all it does. But how it selects and the resulting populations is something I don't think we think about enough in oncology. In general, this sensitive and resistant, one is blood rich, one is a little bit blood poor. The resistant one has to do more with less, and it probably doesn't get as many nutrients. It's an advantage to be this green cell. When there's chemotherapy, it's an advantage to be the green cell. When there is no chemotherapy, it's an advantage to be this purple cell. So prior to diagnosis, months before any pain or mass develops, the sensitive cells that are good at getting blood vessels and sugars and all that stuff grow a ton. And the ones that are squeezed away from the blood vessels grow a lot less. And it's advantageous to be resistant. It's advantageous to be by a blood vessel when there's no chemo, but it's bad with. The umbrella analogy, I don't know that I have time for at the moment. Dr. Gillies is another person in the Cancer Biology and Evolution program, and he writes rather provocative titles like Why Targeted Therapy Does Not Work. But it gets at this point that there are different populations. And even if you had the perfect drug for the majority of cells, a perfect drug against purple cells, all you really do, if there's any green cells, is all you really do is change it so that the purple cells are no longer winning, but the green cell is winning. So you really just expose the resistant cells, and this leads to relapse eventually. So patients love you for these first couple weeks because you're killing cancer cells and the disease is shrinking, but inevitably failure will occur in this case. I'm not talking about osteosarcoma specifically, I'm just talking about general principles of this. What this clearly gets to is more is not always better. Instead, managing these different populations, anticipating how they might get around our chemotherapy becomes the thoughts going forward. And I'll take another pause for discussion.
You mentioned sugar. We get so many people saying sugar feeds the West, what diets do you take? What do you believe? Do you believe people with OS have a sugar issue?
It's a great question. What do I eat? What about sugar? What about vitamins? What about eating healthy? What about antioxidants? All of those things I think are really good questions that are all, I think, addressed with this way of approaching cancer. If I had cancer in my body right now, it is more like me than I am like Anne. Even though Anne and I are both humans, we have at the gene level, we have about 3 out of every 1,000 base pairs is different. My cancer, even crazily mutated, has about 1 out of 1,000 changes that are different than me. I think my cancer would be very much like my body. So I think with your question about sugar, my body needs sugar and cancer needs sugar. I think cancer is better at getting sugar than the rest of my body is because we talked about cancer cells are the unit of selection. They care more about themselves than the whole of me. That's what makes cancer. So it's good at stealing cancer. It's good at stealing sugar off my body. But my body needs sugar. So I would never tell a patient you can't have any sugar. I think that vitamins, the idea that you can give something healthy to the body that wouldn't be healthy to the cancer, I don't know that I believe that. I think that anything healthy for the body is also healthy for the cancer. The cancer also has oxidative stress. It also likes antioxidants. So I don't think mega doses of vitamins or those are going to help the body more than the cancer because the cancer is so much like the body and facing similar stresses. I think that's why chemo sucks. Chemo sucks because in order to kill cancer, you have to hurt normal cells because they're very similar. Unless we can find what's different. That's what's so enticing about targeted therapy and magic bullets is that if you can find something that's only in the cancer, not in the regular cells, you can have no toxicity in that, but that's complicated. And the chemo that works for osteosarcoma has significant side effects. I don't need to tell the panel because you know more than I do. You've gone through it. But I think that's why chemo stinks, is because cancer is like the person's body. One caveat of that whole thing is eating a ton of sugar leads our bodies to naturally make something called insulin to kind of drive that sugar down. And it is true that cancers have some receptors for things like insulin and IGFR and other things. And so I do tell my patients, you know, what Will Ferrell ate as ELF is not an ideal meal for someone with cancer. Spaghetti with syrup, with M&Ms, with whipped cream on it. So very big doses of sugar over a short period of time, high, you know, these glycemic indexes probably is not perfect for cancer cells because they are needing sugar. So giving them a huge dose of it is probably not in the best way.
Dr. Benavente from UC Irvine has a question for you.
Hey, Damon, I'm really liking this view into more philosophical aspects of cancer. I was kind of curious if, in this view of targeting the most rapidly dividing cancer cells, ultimately potentially leading to an advantageous environment for the more aggressive and resistant cells to take over, what are your thoughts on perhaps searching for therapeutic strategies where we keep the cancer at bay and keep it as a chronic disease instead of ultimately aiming for their extinction or the cure for the cancer altogether.
Good, fantastic. I mean, the word that we use here at this Cancer Center for this is adaptive therapy, and Bob Gatenby and others, Jingsong Zhang in the prostate cancer arena have talked about this. It is absolutely a strategy that would come out of this. The most important thing that I need to say about this is, if you cannot cure a patient, I think that's it very well could be the best strategy. It is a very uncomfortable aspect of being an evolutionary oncologist and of what we're talking about today. And thanks for bringing it to the forefront. I didn't know if I was going to address it head on this hour. But if you know that you have a tumor with a whole bunch of purple cells and one green cell, and if you give a drug that's very good at controlling the purple cells, and if it starts to work, it is true that the logical conclusion is to stop the therapy and let the purple cells grow back. And then restart it again, and then let them grow back, and then restart it again and let it grow back. This is a clinical trial with old men with incurable metastatic prostate cancer and a drug called abiraterone. Abiraterone is given during this red time and stopped when these gentlemen's PSA went down. These 11 people went on this study, and some people got abiraterone for a period of time for months, and some people got it for a very short period of time, less than a month. And then they would hold it, and if their PSA dropped, they would hold it, and they would start it again when their PSA went back up. These gentlemen declined the study and said, "What the heck is wrong with you, doctor? You're telling me that when the chemo is working, you're going to stop it? There's no way I will sign up for that study." And all of these people got the chemo continuously. You see with these X's, this is when the patients progressed. You see all of the patients at the same time that went on this study progressed. And one of the patients that did the start-stop worked. If we had a drug for osteosarcoma that worked as well as abiraterone works for prostate cancer, it has a 73% response rate, and many patients have complete responses, but those responses only last for a year typically. And you see the average for these patients was about a year of disease control. Some got better, some got worse, but the average was about a year. And you see with less chemotherapy in this case, and most of the time not being on chemotherapy, a lot of black on these bars, these patients did better than the 12 months. And this study is still ongoing, but this was reported in 2017, I believe. And patients did about twice as good with 44% of the drug, with about half the drug. So yes, Claudia, yes, a thought would be to try to control the disease and use the fast-growing cells to control the resistant cells. The problem with all of these gentlemen down here that got the drug and then now had disease grow through is that there wasn't good options after that. So all you've done is you've exposed the real bad thing, the green cells that were resistant from the beginning. We don't have these drugs perfectly in osteosarcoma, and so when I learned of this concept, the first place we've tried to use it is in frontline metastatic alveolar rhabdomyosarcoma. And so that's the trial that we've just launched to the National Pediatric Cancer Foundation, is start-stop VAC chemotherapy for that rhabdomyosarcoma.
Yeah, Damon, just to follow up, since there's other people around, how feasible do you see it? Because in prostate cancer, you're talking about fairly old man, and how the feasibility of doing this in the young population and potentially having to go on and off for the rest of their lives. Have you ever thought of like potential, how non-toxic the therapeutic has to be for it to be a plausible option?
So it's outside of the scope of osteosarcoma. So I don't know how to do it in osteosarcoma, so I haven't proposed that yet. I don't think that there's an adaptive approach to osteosarcoma that makes sense right now. I don't think we have agents that for lack of a better word, work that well. I don't think we have those kind of response rates to do that. How feasible it is will be studied with that rhabdomyosarcoma study. So that it's going to have 4 options for care. One of them being more aggressive chemo, one being early maintenance. These are contextualized in the eco-evolutionary framework as being a stronger first strike and a second strike, as many extinctions of animals have happened not because of an asteroid, but because of small subtle changes in the habitat after. So these are principles that I don't know that we can get to in this time frame, but there is an adaptive arm for newly diagnosed fusion-positive rhabdomyosarcoma patients. This is not a randomized study, the rhabdomyosarcoma one, and so patients and physicians will decide. And so we'll see if people choose the adaptive arm. So what are we doing at Moffitt with this information and this strategy? One is we have an evolutionary tumor board, co-led on the clinical side by Dr. Christine Chung, and also led on the IMO side by Sandy Anderson. We get together, Christine Chung, head of the chair of our head and neck program, brilliant, and myself as the clinical leads, and Drs. Anderson, Gatenby, Brown, and many other designees, dozens of them. We think about patients this way. So we measure, we consent patients that are incurable, and we present an evolutionary tumor board. We get into incredible detail and think about things like, should we give strong chemotherapy? I've recently given a patient with relapsed sarcoma frontline therapy again years later to try to see if that would help. So far, it's not doing bad, actually. We talk about second strikes, so hitting it, kicking the cancer once it's down. We talk about adaptive therapy, so start-stop therapy, trying to trick tumors into double binds. And what we're doing with this evolutionary tumor board is presenting patients and seeing if the mathematical oncology group can come up with different ideas than what experienced clinicians would do. So the patients look something like this, and it takes about 6 weeks to get all of the information together for one patient. And so this patient received some therapies, got radiation, and this is how much disease they had. They had no evidence of disease. This next therapy, what responded, what tests were done, what genomics were done, and what surgery, radiation, all these combinations of agents. And then this patient was a patient that was explored retrospectively to try to see if we could have done something different, but unfortunately had already passed. But we were discussing the dynamics of how these different strategies worked. And we're trying to work, and this is a lung cancer patient, trying to work on how this may have been different. And so lots of people thinking about patients in a different way with lots of different disciplines, and that's our evolutionary tumor board. This same group, Sandy Anderson, and then Mark Robertson-Tessie, brilliant person in Sandy's lab, and Andre Marusk, a brilliant experimental biologist that studied some of these concepts. We ideated on osteosarcoma for a week pre-COVID when people could all get in the same room, and we invited graduate students from around the world to come and learn about modeling. We took a couple patients' scans, and we really just let these brilliant people kind of go. And they conceived that there is a birth and death rate of the primary tumor and a birth and death rate of metastases. And there's a certain detection threshold on CT scans. If our CT scans were perfect, you would get one CT scan and you would never need one again, right? Because you didn't have disease, why look there again? But our CTs could look down to a certain size. What they showed is that there are certain sizes of lesions that could, with very effective therapy, just disappear and then come back. They showed that I was thinking that from a clinical aspect that they were going to tell us when the best time to do surgery was. But what ended up coming out of this was that there is actually the worst kind of lesion is the one you can't see from the beginning, which I think we all know. What you can't see is troublesome. But through math, they were able to kind of show and suggest different ways that we would go about treating osteosarcoma. And so we're still working now that we have a mathematical model, now that we have some patient data fed into it. Now Andre is working on mice to try to see if some of these strategies will hold for better ways to manage osteosarcoma. And that's the whole group of people that participated in this. I had a beard back then and a little bit more of a belly. So that's one thing that we're doing, evolutionary tumor board and working with the integrated mathematical oncology thing. I think that another thing is, and we'll, with time, name some genes in osteosarcoma. And some people would yell at P53 tumor suppressors, which are generally lost in osteosarcoma. So they let the cancer these cells grow out of control. There are oncogenes like MYC, CDK4, and some others, PI3 kinase, RAS, that are increased in osteosarcoma. And then there are housekeeping and wild type genes that are housekeeping and wild that are unchanged. Primarily, people talk about P53, but oh, alas, it's undruggable unless maybe elephant P53 or something cool like that could happen. Oncogenes are increased, but unfortunately they have normal roles in healthy cells. So even if we had perfect drugs to knock them down, we're going to have toxicity because our normal cells need oncogenes too. MIC has normal roles in our body. You can't completely block it. You would have too much toxicity. But everyone throws away these housekeeping wild type genes. They're unchanged. They're never changed in osteosarcoma. I think one of the things about evolution is that it says that's important too. It's not only important, it doesn't only select for gains or losses of DNA, it also selects for it staying the same. So you've seen crazy genetics of osteosarcoma. Each one's a snowflake, each one's different, each one has chaos in the genome, lots of gains, lots of losses. We have taken this data and focused on what never changes, rather than look at, like, say, what Alejandro, who's brilliant, he looks at what's gone up and tries to knock it down. Again, a paper, 2014, same group, Gatenby and Brown. Evolutionary Triage governs fitness in driver and passenger mutations and suggests targeting never mutations, that the most effect is by actually weakening these. And to me, when I really bought into this idea about 4 years ago, I thought, what is the most central function a cancer must never lose? And it has to be able to make more DNA. You can't have, if a cancer cell can't make another cancer cell, it's not cancer, right? So what makes DNA? Fortunately, we have a guy named Mark Alexandro here that has studied the CMG replicative helicase over and over and over again, and he's been studying it for decades. And this is what wraps around DNA, splits it, and adds new C's, T's, A's, and G's to make another cell. This is never mutated in any cancer, because if you lose the replicative helicase, you're not cancer. You can't make DNA. This is a never mutated gene. And so Dr. Alexandro has an idea that this ATP pocket could be druggable. We could find a drug to attack this. And he has shown tons of data that cancer cells are relatively vulnerable to targeting that ATP. So rather than a situation where a cancer cell has more of something, a normal cell has this much, and you give a drug and you get toxicity because you knock down both, the idea is the opposite. A cancer cell is living on the edge. It has just enough CMG helicase to make DNA. Our normal cells have extra backups. And so a drug could actually selectively knock out osteosarcoma and the normal cells wouldn't even shrug, is the concept behind this. Very early, he's still working on making these huge proteins in a way that we can try to make the ATP assay go. And I won't say that's incorrect. His lab is doing this towards trying to figure out drugs for this. Just to make it more exciting for an osteosarcoma audience, there's some data that he's published that two proteins interact with this. Those two proteins are genes that everyone in the osteosarcoma should be aware of. One is MYC, and the other is RB1. Both seem to interact around here with this CMG helicase. RB1 holds it back, and we lose it in osteosarcoma, and so it can make more DNA. MYC stimulates this complex. And so you can see how that would be bad for faster cancer cell growth. And him and I wrote a paper, MYC and the replicative CMG helicase, the creation and destruction of cancer. Good title, but it's a good read if you really want to nerd out. Again, these are never mutated. This is 40,000 patients, 42,000 samples through CBioPortal, and these are they excluded the ones that are of unknown significance. So there's a little bit of an asterisk here, I'll be honest, but 0% mutated. And again, some rationale for why tumor cells would be selectively more vulnerable. So that's one thing we're working on. We're working with a former yeast biologist, a person who studies DNA replication in detail and is trying to apply that to cancer and has been for decades. A very experienced, very good researcher, and it's been a very exciting collaboration. We have knocked down some CMG and some osteosarcoma cell lines, and we have indeed showed that they are vulnerable to chemotherapy. Particularly, we've looked at topotecan and doxorubicin, and we've shown that they're more sensitive to that chemotherapy if you do artificially knock down that CMG helicase. Connor Lynch, Jeremy's been he went to the first MIB Agents, loved it, really got into osteosarcoma, and that directed this project, actually, got him interested. And Jeremy's published recently that histone deacetylase inhibitors, and he works in Connor Lynch's lab, and now Nivedisa is taking over this project as a grad student. I just submitted a grant recently to the federal government for this project. So we've studied panabinostat, an HDAC inhibitor that seems to have really good activity, not only on the dish and the plate of osteosarcoma cells, but also in the mouse through Jeremy's work. Nivedita is trying to figure out why. These HDAC inhibitors like panavitistat do a lot of different things, and she's focused on a protein called SLC17A7, a glutamine transporter. And so there's a lot of people that are working on glutamine, and so we like the idea of potential synergy with smart people like Amy LeBlanc and others to try to see if we can go down this road. My own personal lab, the Sunshine Lab, supported by the National Pediatric Cancer Foundation, we take a lot of RNA-seq data, a lot of data of how sensitive these cell lines are to different chemotherapies, and we explore how we can compete cells against each other on a plate, and we're, again, exploring good combinations to attack this CMG helicase. We study growth over time, we change the conditions, we decrease glucose, as was mentioned before, glutamine, and what we see is that a cancer cell that dominates with plenty does terrible with little. And so we think we have our prototypical green and purple cells, 143B and SOUS2, and now we're working on making assays to kind of compete them against each other to show experimental evidence for the theoretical stuff that I talked about in the beginning. And then also, we believe that just like we talked about with the IMO workshop, CT scans lack the real threshold of activity that we need to see, the sensitivity to these small populations of osteosarcoma. And so again, teaming with Dan Weiser, Masanori Hayashi, Mike Isaacoff, and David Loeb, we have this protocol, the Raigai Foundation, and Rally is also supporting this along with National Pediatric Cancer Foundation. We're drawing blood in osteosarcoma patients would be on this arm. They would get blood drawn at diagnosis, a couple weeks after cycle 1, prior to surgery, after surgery, 2 weeks after surgery, end of therapy, and during scans to try to see if we can detect osteosarcoma more sensitively with the blood than we can with scans. We hope that learning about how fast it goes down and how low it goes and how long it stays down, we think all of these might be better ways to anticipate relapse, or maybe find out if therapy is working wonderfully or we need to switch therapy earlier. But this is all over the next 3 to 4 to 5 years, collecting these samples, learning about how patients did. We're not returning these results to patients or the doctors. This is all happening. And at the end, we're going to look and analyze to try to see how useful these blood tests are to help future trial design and management of osteosarcoma. But I'm very excited about this several $1 million project. In summary, cancer adapts. And in general, I'd like to commit in trial design to 3 to 6 week cycles, not 29-week plans. I'd like to be able to figure out how our chemo worked or surgery worked. I'd love to have a blood test or some way to figure out exactly what's going on. Then I'd like to rethink about the patient and kind of, like a wolf, kind of anticipate its next move. Did chemo work really well? Great, let's continue that. Did chemo work okay? Is there something better we can think about? I think we're not one intervention away from a fix. So while we still do conduct some studies that are MAP plus something, or, and I think there are some good ideas like tyrosine kinase inhibitors, I do think that a trial design where we're doing more than one thing tends to be very easily welcomed by the advocate community. And so I think that we need to continue to think how we can make trials as potentially beneficial to patients as possible. I think words matter. It was brought up earlier by Claudia that adaptive therapy. I think if I sit there and I just say things like works, oh yeah, this chemo works to a patient, and I feel in my heart of hearts there's no way I'm going to cure them, I think that we have to think about the goals of care, and at times things like starting and stopping chemotherapy may make more sense, not only for toxicity, but maybe patients could live longer by giving less chemotherapy, is something that is one of the most uncomfortable but really logical aspects of thinking of cancer this way. I don't use the word standard of care too much. I think in osteosarcoma we have things that we do. I would call them conventional chemotherapy, but I don't think they work well enough that we would call them a standard of care. When the outcome is more than 90% of people live, I'll call it a standard of care, but that's my threshold. I call everything else conventional chemotherapy, because I think when we call it a standard of care, it seems like it's non-malleable. We can't change it. Again, I think goals matter in care, and I think there's tons of opportunities in trial design. I think there's opportunities to personalize schedule. I think people that respond faster maybe should get different chemotherapy than others, so I don't have good enough data to do that in osteosarcoma today. I think we should anticipate and have resistant management plans. This is Joel Brown's idea, to try to keep those green cells from escaping. I think we should measure perturbations. How did we change the tumor? Not necessarily just by size, but how did we change it? Is it more metabolically not active? Has it done this? Has it done that? Does it have more blood vessels or less blood vessels? And then we can start to think about how we might go about the next strike, the next thing that we would do. And I also think it's really cool to invite other disciplines into the osteosarcoma field. I got a hint. I think I know some stuff, but I definitely felt at a moment that someone outside of oncology might have some good ideas. And that was when I got to meet Joel. And as a squirrel biologist, he has a bazillion good ideas. And I think how to eliminate pests and invasive species has been more informative to me than what's the mutation, where's the drug that fits into that pocket, and let's give it to the maximum dose until people feel sick. So I think that there's a wonderful team at Moffitt that thinks of things differently. Not that different is always best, but I've certainly taken the hook and really enjoy working with all the members here that I mentioned throughout, but led by Bob Gatenby, Joel Brown, Bob Gillies, Elsa Flores, terrific mentors here and terrific scientists, Mark Alexandro, Elsa again, Connor Lynch, many others, Andre as well, Mark Robertson-Tessie. A lot of people are paying attention to osteosarcoma here, and we've got a lot of supportive funding. It's really been fantastic, and thanks for letting me share some of it.
Thanks for sharing it. And most of all, thanks to you and the team. Really, really encouraged and enthusiastic about Moffitt's commitment to osteosarcoma. We so often feel like we're the forgotten cancer because we've not seen any new treatments or new developments, and this is really hopeful. Oscar Ortiz is present, and he has a question for you.
Early diagnosis is so critical to long-term success. As we went through what we went through with Sebastian, we recognized had it been caught earlier, our chances would have been so much better. And that's probably likely in so many cancers. And a lot of the advances, it seems like, in adult cancers have come from, as much as treatments, diagnostic tools that allow for pre-symptomatic diagnosis. Now, knowing that it's more challenging for kids for many reasons, smaller sample sizes, divided up amongst a variety of different cancers. Are there, and you mentioned some of the testing you're doing for the blood instead of scans, are there any tools on the horizon that would assist with sort of early diagnosis, pre-symptomatic diagnosis for kids in the fight or for any type of cancer in the pediatric space?
It is, the challenges are many, and you hit upon them. Having a rare event happen in a population is already like a couple strikes against any kind of screening kind of assay. But these sorts of blood tests would be a potential path forward. I think that we already see that colonoscopy is not the only option for screening for colon cancer now. So now you see that little box of toilet paper that is advertised on TVs, and so now you can send a small piece of stool or a piece of toilet paper, and they can try to screen for colon cancer. And that Cologuard works almost as well as a colonoscopy for the right patients. And so I think that there are massive efforts led by some of the massive information companies, Grail and others, by like Google and others, that are trying to detect cancers in adult populations where it is both more common. And I do think that something like that can be in the future. Certainly people are going that way. And I think that we will definitely see on our own yearly physicals getting blood tests to screen for cancer before we see it in pediatrics. But once the principle is done that you can detect cancers early that way, then I think that there's no reason why it should be different for pediatrics. I will say the first 2 weeks of medical school taught us the math and the statistics for why BRCA1 screening in breast cancer was so complex when I was in medical school in 1998, because of the percentage and because of that. We will be dealing with all numbers that are more unfavorable as we try to screen for cancers in kids, but it doesn't mean people can't try.
What data would help you personalize or anticipate care? Genetics, scans?
I would say that the answer for me would be scans. The answer for me would be if we're thinking of cancer as a population of cells, then volume measurements of all disease would be super helpful, because then you can calculate an approximate number of cells. You could get an idea of how many cells there were on this stage and how many cells there were on this stage, and you can calculate growth kinetics, how fast it's growing. You can, that's one thing that we get on all of our evolutionary tumor board patients. You can see that cells are growing at 2% per day. And when you give cancer, you're reducing them by 5% per day. Those sorts of growth kinetics are super helpful, because a lot of times patients go on something and they say, I don't know, what would it have been like if I wasn't on anything? And if you had volumetric measurements over time, you would have a better sense of what tumors are. Right now, we rather lazily measure a couple lesions on patients. So we might measure up to 5 in one dimension. Those numbers are pretty useless for trying to see how much disease a patient really has. If we had a huge database of osteosarcoma patients from diagnosis through relapse through lots of therapies, I think we could get a much better sense of how much therapy bend the curve. And I think then you could start thinking about how you might stagger them to maximize patient benefit. It's weird though, it's unintuitive. The more a patient responds, if you cannot get rid of all the disease, the more likely you should be to stop it early. And that is a weird thing to think about, but that's the principle of adaptive therapy.
All right, so let's wrap this up by saying Childhood Cancer Awareness Month, also known as September, has 2 more great sessions and guests. Next week, we have Dr. Brian Flesner, speaking of doing things a little bit differently. He's from the University of Missouri. He's a veterinary medicine doctor that specializes in cancer. He is with the University of Missouri. He'll be speaking on adjuvant chemotherapy for canine osteosarcoma. And the following week, we'll have CCTDI team members, Dr. Charles Keller and Andy Woods, who'll be talking about legacy autopsy gifts. So important. Nobody talks about this, but when a child dies from cancer, that their data, their body is so important to research. So we'll be talking about the afterlife gift of tissue, which is funded for osteosarcoma patients entirely by our friends at the Childhood Cancer Project. So important. Finally, the most comfortable t-shirt ever, right here. It can be yours too, along with the cape, which also super cool right there. When you outrun osteosarcoma with us, you can sign up on our website on MIBagents.org under what we do, or email us at info@MIBagents.org for the link. We'd love it if you signed up, and love it even more if you asked your friends and family to sign up too, in honor of an OsteoWarrior. Your participation really allows us to continue to support research and all of our programs. So thanks for joining us today. And of course, thanks to our guests, Dr. Damon Reed, and our panelists, Ryan and Amanda. Together, we make it better for osteosarcoma kids everywhere. Wear your mask, please. We'll see you next week. Sign up on our YouTube channel for early viewing each week of new videos and access to the entire library of OsteoBites. For OsteoBites podcasts, listen wherever you get your podcasts. Until next Thursday, please use your powers for good. For kids with osteosarcoma, visit our website at MIBagents.org.